right, this is the protein lecture. So first, let's go through an overview of what proteins are. Hopefully you remember from Principles of Nutrition that we have dispensable and indispensable proteins. These are sometimes also called essential and non-essential. The difference between these two are if we can make them. If we can make the protein, it's non-essential. If it's essential, we can't make it, and we need to consume it. So hopefully these essentials look familiar. I want to draw your attention to the conditionally essential. These are amino acids that we make, but in times of stress, we may not be able to make enough. Uh, so these become essential based on stress on our physiology. And this isn't just psychological stress. This is usually something like um, having a severe burn. So, another reminder of amino acid structure. Every amino acid follows this structure. We have an alpha amino group, which is attached to the alpha carbon. This alpha carbon also has an alpha hydrogen. If I were to draw this, I would have put the hydrogen here. And then this carbon is attached to this carboxyl group. And you'll notice this carboxyl group has a negative charge, and this amino group has a slight positive charge. So this structure here is common to all amino acids. The R group or the side chain, though, is unique to the amino acid. And this is what gives the amino acid its character. And you may be wondering, well, what do you mean by character? Oops. Also, this is the acidic part. So here we're looking at some of the different amino acids, and we have different types or groups of side chains. So here we have electrically charged side chains. So you'll see that we have a positive charge on arginine. There's a slight positive charge on histidine between on this group here. And there's a slight positive charge here on lysine. We also have negatively charged. So aspartic acid and glutamic acid have these slight negative charges. Or not slight, they have a negative charge. So these side chains are going to interact a lot with other side chains. With side chains of opposite polarity or charge. So recall that water, H2O, is charged. Well, it's polar. It has a slight positive charge on one side, and towards the oxygen, a slight negative charge. You'll notice when you have a bunch of oxygens, you tend to have a slight negative charge because they are electron hogs. So the electrons are hanging out next to oxygen. They're not hanging out as close to the hydrogens. So there are also amino acids with polar side chains. If something has a polar side chain, it is going to interact with water. It's usually hydrophilic. And so you'll see the polar side chains here. We just have an alcohol group. Here we have an alcohol group and a methyl group. Spargenine, you'll see, looks quite similar to aspartic acid, only a different nitrogen group here, and also glutamine, which we have here. We also have amino acids that have hydrophobic side chains. So things that are hydrophobic, think fats. What does a fat look like? Well, it has a glycerol backbone, but then it has a tail made up of carbons and hydrogens. This part is hydrophobic. We don't see any oxygens in here. This tail does not want to interact with water. And in fact, this is why we get micelles forming. Think about the micelle are usually some form of lipids. We'll say they're a phospholipid here, and they turn their polar heads outwards, and they put their hydrophobic tails in the center. These amino acids are very similar. So you'll see here, this is just a methyl group on alanine. Very simple. On valine, we have two methyl groups. Isoleucine, again, just carbons and hydrogens. Leucine, just carbons and hydrogens. Methionine is interesting because it does have a sulfur, although this is still hydrophobic. Phenylalanine, all carbons and hydrogens, hydrophobic. Tyrosine, this ring is hydrophobic, although this OH group is slightly polar. And then this group here is hydrophobic. I do want to draw your attention to valine, isoleucine, and leucine. These are, the branch chain amino acids. You've probably seen these in GNC. The reason they're called branch chain amino acids is because their R groups look like carbohydrates or like fats. They're just carbons and hydrogens. And that means that we can break down branch chain amino acids for energy. And in particular, the muscles can use them for energy. Methionine is also important because methionine is critical for the SAM-SAW pathway. And you may remember from biochem or something like that, the SAM-SAW pathway is going to be important for DNA methylation. And it depends on folate, B12, and B6. So let's remind ourselves how we make proteins. this is inside a cell. First, we have to take the DNA and we have to transcribe it to RNA. So we're just making the DNA into a copy RNA. So T becomes U, and then we make it a messenger RNA. This is just tagging it and allowing it to leave the nucleus. And then we translate the mRNA into a protein. So here we see transcription. Topoisomerase and the other enzymes have unzipped the DNA. The RNA is being transcribed. The RNA gets tagged and is allowed to leave a pore of the nucleus, and this is the mRNA. The mRNA then has to go to a ribosome, and a ribosome has a large subunit and a small subunit. And essentially, ribosomes are protein-making factories. Or I sometimes think of them as a workbench. And remember, ribosomes are primarily in the endoplasmic reticulum. And they're what makes it rough looking. So our mRNA goes to our ribosome. Our ribosome forms in its two units together, and it then recruits tRNA, translational RNA. The translational RNA has a set of codons, a set of 3 codons, that are unique to each amino acid. And these codons test, they see if they match up with the codon with the mRNA. If they match, they attach, and the mRNA moves down, and the next one comes through. If it matches, it attaches, and then we get a bond between the carboxyl and amino groups, and we get the formation of a protein chain. And that brings us to the primary structure. So here we have our R groups. We have an amino group attached to a carboxylic. Here's our alpha carbon, and here's our next amino, R group. I like to think of the primary structure as beads. It's just amino acids on a string, like a string of beads. But recall that based on the R groups, these R groups are going to interact with each other. And by interacting with each other, either by polarity, non-polarity, or charge, they're going to fold. So if 2 R groups right next to each other have the same polarity, they're going to repel each other. If they have opposite polarity, they're going to pull towards each other. This pushing and pulling due to the R groups forms the secondary structure. The secondary structure is formed through these hydrogen bonds. If you don't remember this, go back to your OChem notes. So hydrogen bonds are less stable than, say, a covalent or ionic bond, but they form these somewhat flexible bonds between R groups. Or between, say, the hydrogen on one alpha carbon and a carboxylic group on a different. So they can interact on their R group, they can also interact on this chain itself. And there's two classical shapes. Often, proteins will create an alpha helix structure, or they'll create a beta pleated sheet. And again, this just depends on the R groups. When we have multiple secondary structures, so you can have multiple alpha helixes, the alpha helix, it can be an alpha helix in one part and a beta pleated sheet in a different part, you get the tertiary structure. So here we see, and this is actually a subunit of hemoglobin, that this subunit has an alpha helix, and it has, it's not shown very well, but a beta pleated sheet. So this is made up of two secondary structures, or actually the beta polypeptides here, but they don't show it very well. So this helix will interact with the beta pleated sheet, again, through hydrogen bonds and folding. When we have two or more proteins interacting, we have the quaternary structure. And see here we have the 4 subunits of hemoglobin, 2 in green, 2 in orange. These hemes are what is going to hold iron and attract oxygen. And so this is 2 or more proteins that create the final form. So you're probably familiar with the fact that in our diet, we have to consume proteins in ideal ratios. And we can quantify this through the protein digestibility corrected amino acid score. The highest possible score is 1, or 100%. And that means there's 100% of the indispensable, or essential, amino acids per unit of protein. And a perfect protein on this scale is an egg white. The digestible indispensable amino acid score is similar, but it allows for values greater than 1 because it's also taking into account the digestibility. So we use proteins for almost everything. They're enzymes, hormones, they form structure. So let's think some enzymes we know of. Hexokinase is a protein. G3P, not G3P, G3P is a carbohydrate, but hemoglobin, nope, not an enzyme either, that's a structural. Gosh, I'm trying to think of a non-kinase, but phosphofructokinase. And tons upon tons more. Lactase, there we go. Hormones, most of our hormones are peptide hormones or protein hormones. So that includes insulin, glucagon, glucagon-like peptide 1, CCK. Really, most of the hormones that we have are protein hormones with only a few fat-soluble hormones. Some structural proteins, keratin, muscle, collagen. We also have our immunoproteins. These are part of our immune system complement. Transport proteins like albumin or retinol binding protein, and many others. Albumin is also important for fluid balance, connective tissue, as well as a biological buffer, which albumin also does. So, an overview of protein digestion. As the digestibility protein index hinted at, essential amino acids are absorbed faster than non-essential. And branched chain amino acids are absorbed faster than smaller amino acids. And this should make sense because these can be used for energy. The absorbed proteins can be shuttled to the liver or used inside the intestinal cells. And once in the liver is really our center of making new proteins. It can be used for a variety of physiological uses. We can synthesize enzymes, we can synthesize hormones, we can synthesize nitrogen containing compounds like urea. So say we eat a steak or tofu steak, and we chew it up. It gets to the to our stomach. We get gastric secretions, including gastric and pancreatic proteases and peptidases. And then our proteins are broken down primarily into di and tripeptides, meaning there's a group of either 3 or 2, or single amino acids. And then there are some large peptides that might still be intact, but mostly it's going to be di and tripeptides and amino acids. These large peptides actually have to either be broken down into single amino acids or di or tripeptides to get into the cell. Once in the cell, we can break down the di and tripeptides even further into amino acids, and that's mostly what we have going into circulation. We have very few di and tripeptides going into circulation. So proteins are broken down into amino acid and oligopeptides, meaning 2 to 10 units. The intestinal enzymes finish the digestion process. So these are enzymes that are embedded in the cell membrane that finish digestion. Individual amino acids, di and tripeptides, are absorbed through the wall of the small intestine. Most are taken to the liver via the portal vein. But again, we have our special ones. Branched chain amino acids may bypass the liver and go directly to the muscle after absorption, but this is a very small amount. So as an overview, we have some protein-containing food. Let's say we ate a bunch of chickpeas. First, we have pepsinogen, and hopefully you recall that if it has a gin on the end, it's inactive. I'm going to change the color of my pen because it's hard to see it with the orange. We have stomach acid, hydrochloric acid, and that's going to activate pepsinogen into pepsin. Active. Pepsin is going to break down the protein into polypeptides, amino acids, and some intact proteins. And all of this is taking place in the stomach. Sorry, I wrote over that. This is in the stomach. So these polypeptides signal for the secretion of CCK and secretin, or secretin. And especially secretin is going to signal to the pancreas to produce proenzymes. Pro meaning, again, inactive. Think about why we need inactive forms of protein enzymes. We're mainly made up of proteins. We're basically protein containers filled with water and bacteria. So if we just allowed our protein breaking down enzymes to run free, we would digest ourselves. So we need to be very careful and very purposeful with our pancreatic proenzymes. And these are things like trypsin, chymotrypsinogen, and these will further break down the polypeptides and intact proteins into even more amino acids and oligoproteins, oligopeptides. These will then interact with the brush border intestinal enzymes, and we get brush border absorption. This is taking place in the intestine, and CCK and secretin also from the small intestine. And from there, our amino acids and some tripeptides go to the liver via the portal vein. But this class is really about weight control. So how does protein affect weight control? The most obvious is the thermic effect of food. Dietary protein requires 20 to 30% of its usable energy to be expended for its own metabolism. This is inefficient, which means we're getting less energy per protein because we have to spend 20 to 30% to digest it. Just think about all the enzymes. On the other hand, carbohydrates require 5 to 10%, and dietary fats, these are easy. These are highly efficient. And they require 0 to 3% of their own energy to be metabolized. So putting on fat and eating fat is very efficient. Burning up the food that we're eating to metabolize the food is inefficient. But the thermic effect of food probably contributes very little to weight maintenance. Though we do know that higher protein diets prevent a decline in resting energy expenditure during calorie restriction. But part of this is probably from muscle sparing. You may have heard that protein-rich meals make you feel fuller for longer. And there have been studies showing a modest satiety effect with protein-rich meals. Unfortunately, this satiety, which is the time between meals, doesn't affect energy intake at the next meal. So we may wait longer to eat, but we don't eat less at the next meal. There is a protein-specific appetite in that we need to consume proteins. So it is potentially to make sure that we eat enough protein, but not too much protein. But there's also non-specific appetite. Protein triggers the release of PYY and GLP-1, which we'll get to more in the endocrine lectures. And these are associated with satiety and satiation, which we'll also get into more. Hopefully you watched the video on forest plots. If not, please watch the video on forest plots. But briefly, this is a large forest plot. And we see that it actually has a couple of different things going on. So first let's highlight. It has an intervention category. So this first one is protein versus digestible carbohydrates. The next is protein versus fiber. You can tell this is from a British journal because they spell fiber different. This one is protein versus fat, and this is protein supplementation versus no supplementation. So we see here that we have a lot of studies looking at protein versus carbohydrates. But we have very few studies, just two, looking at protein versus fiber, and only two looking at protein versus fat. So within each of these groups, we see that there's a subtotal I squared with a p-value associated with it. And we have an overall total down here. And these totals are associated with these diamonds. The diamond indicate the ends of the diamonds are the confidence interval. The middle of the diamond is the mean. And in this case, we're looking at a difference of means. So if mean 2 minus mean 1 equals 0, that means mean 2 and mean 1 are the same. But if it's greater than or less than 0, which we see down here, it means that these two means are different. And we quantify this by saying if the confidence interval contains 0, so CI contains 0, there's no difference between means. So here we see that overall, there is a slight effect of protein versus other foods. But if we look at the individual ones, like the protein versus fat, there's no difference. You can see that this diamond crosses 0. If we look at the protein versus fiber, it's hard to see, but it's also not significant. Really, only protein versus carbohydrates is significant. And if we look at protein versus carbohydrates and the overall, you'll see that they're very similar. So this effect that we see here is due to carbohydrate studies. So this is saying that protein is more satiating than carbohydrate, but not more satiating than fiber or fat. The other reason that we want to pay attention to protein is because protein builds muscles. And skeletal muscle is normally responsible for more than 75% of all insulin-mediated glucose disposal. So go back and remember how skeletal muscle contributes to glycemia. So given the relative size of whole body skeletal muscle mass, which is quite a bit, skeletal muscle is a key tissue in whole body glucose homeostasis. More muscle equals better glucose disposal. Muscle is also a major site of lipid oxidation, where it has a unique role in modulating whole fat balance via aerobic metabolism. So a lot of the skeletal muscle is going to do first just glycolysis. And we can only do glycolysis with carbohydrates. This is for our fast twitch muscles. But as we run out of glycogen, we need to use fats. To use fats, we need aerobic metabolism. And then remember, aerobic metabolism means it requires more oxygen. So it's going to create a or equal an RQ around 0.7 because it needs more oxygen. And insulin is an anabolic hormone. We usually think of testosterone or DHEA as our anabolic hormones, but insulin is highly anabolic and it promotes muscle synthesis along with lipid synthesis and also glycogen storage. Well, lipid storage, I guess, is a better way of saying it. On the other hand, glucagon, cortisol, and adrenaline all promote muscle loss. So these act against or in converse to insulin. So the effect of protein on fat-free mass. Protein has been shown to help spare fat-free mass during weight loss interventions. And this is really important. Remember resting energy expenditure. Go back to those notes and think about how body composition can affect our EE. Also think about that when we lose weight, we're usually losing water weight, fat-free mass, and then some fat mass. And also consider the low fat versus low CHO diets we just talked about at the end of the carbohydrate lecture. So we know that we're going to lose muscle during weight loss. And this is largely due to using our own muscles through gluconeogenesis. So consuming protein will help prevent the use of our fat-free mass. And this is more effective if the weight loss intervention incorporates resistance exercises. This is how we are going to build muscle. So here we have a table showing anthropometric, biochemical, and dietary intake during an energy restriction with 2 levels of protein intake. We have normal protein, higher protein, and then this is the comparison. This also took place at baseline and 6 months. So let's look at what is significantly different between time and diet, because that's the most important. We want to see the effect of the diet over time. And there's only a couple. Obviously, the amount of protein per day is going to be different. That's also protein or grams per kilogram. So that's our diet manipulation. But then we also see that blood urea nitrogen is different. So let's look at baseline then. Normal protein baseline, around 13. Higher protein baseline, slightly lower. But not a huge difference. At 6 months, though, we see that in the normal protein diet, we've seen a decrease in blood urea nitrogen. But we actually see an increase in the higher protein diet, which makes sense because we're consuming more protein. But if we look at things like changes in weight, no difference between groups over time. And in fact, no difference between groups overall. What they did find, though, is that high protein diets decrease gluconeogenic precursors. Remember that gluconeogenic precursors are proteins. So again, we have our normal protein and we have our higher protein. And they broke down their amino acids into different categories. The glucogenic, so it can be made into glucose, the branched chained. And if we look at this difference, these are all different by time and diet. So we see that for the gluconeogenic enzymes in the normal protein group, they increase. In the high protein group, though, they decrease. So think about which group is using their muscle. If we look at the branched chain, we see a slight decrease. But in the high protein diet, we actually see an increase. And this is interesting because these can be used as fuel for muscle. So what this is showing is that a high protein, I'll just erase that. A high protein diet, so we're still in negative energy balance, is going to spare muscle. And it's doing this by decreasing gluconeogenesis using muscle. If we look at the serum amino acid concentrations, again, same manipulation, normal protein versus higher protein, we see that some specific amino acids are different. And the one I really want to draw your attention to here is alanine. Alanine was different by time and diet. We also saw differences in glutamine, glycine, and lysine. with lysine being a branched chain amino acid, much higher. Glycine, not a branched chain amino acid. So glycine's actually higher in the normal protein group. And alanine is slightly higher in the high protein group. I mean, slightly higher, but the percent change is much, much less. So this is indicating that less alanine is being used because the percent change is negative. And it is much more negative than the percent change here. So this then, it helps if we think about where do we use different amino acids in gluconeogenesis and in the TCA cycle. So the red ones are gluconeogenic, the green ones are ketogenic, and the purple ones are gluco and ketogenic. So notice alanine and glycine, both glucogenic. They can enter this pathway as pyruvate. Aspartate, asparginine, can enter as oxaloacetate. Aspartate, along with tyrosine and phenylalanine, can enter as fumarate. Threonine, methionine, isoleucine, and valine can enter at the level of succinyl-CoA. Alpha-ketoglutarate and glutamate can go back and forth. But arginine, proline, histidine, and glutamine can then become glutamate. And finally, tryptophan, tyrosine, phenylalanine, threonine, isoleucine, lysine, and leucine can all come in at acetyl-CoA. And really, this is dependent on what the R chain looks like. So with this protein precursor, first we have, this is the TCA cycle. If we go up this way, this is gluconeogenesis. If we're going this way, it's glycolysis. But especially at the level of alpha-ketoglutarate, we can cycle through glutamate. We can make glutamine or use glutamine. We also can use this to get rid of excess nitrogen through the urea cycle. We also have the Cahill cycle. And this is important because this is a cycle that's going to cycle, saying cycle a lot, proteins between muscle and the liver through blood. And in fact, it has two main parts. It's cycling alanine and glucose. So alanine gets pulled out of the muscle, gets made into pyruvate. Pyruvate gets made into glucose. This is gluconeogenesis. And then we now have glucose. Glucose goes into the blood. The muscle needs glucose. It's going to do glycolysis to pyruvate. And that pyruvate can then be turned back into alanine and the cycle continues. You'll notice that this is anaerobic because this pyruvate could go to a TCA cycle, but instead is being turned into alanine, and then alanine is being used to make more glucose. So we're actually making less ATP, but we're maintaining blood glucose. So according to Cahill and the co-authors, who the cycle is named after, of the 20 amino acids measured, alanine is the principal amino acid released by the forearm, in accordance with its being the principal amino acid extracted by the liver for gluconeogenesis. So alanine comes from our muscle. It goes to the liver for gluconeogenesis. And this occurs in the post-absorptive state, aka fasting, and after 4 to 6 weeks of starvation. After that, amino acid release is markedly diminished. Basically, we start to run out of muscle. Alanine provides around 3.425 kcals per gram, and it accounts for more than a quarter of the total amino acids taken up by the liver from circulation. So in summary, protein tends to have a slight satiating effect compared to fiber, but this is pretty weak, and carbohydrates. This is pretty strong. Low-fat diets tend to spare muscle. Reread the end of the CHO lecture notes. This is because less protein is needed for gluconeogenesis because it's low fat, so we're consuming enough carbohydrates. And protein appears to protect lean, whoops. Oh, shoot. Sorry. There we go. Protein appears to protect lean mass during weight loss and may increase the efficacy of weight loss overall. So think about the effect of resting metabolic rate or resting energy expenditure. I guarantee I will ask quiz questions about that. And that is the end of the protein lecture.